Hello, everyone, and welcome to Professional Previews. My name is Michael Lewis, and I'll be your host today. And today we are joined by Dr. Carlos Goller, who is a professor, associate professor at NC State, assistant professor.
Uh,
And so,
yeah,
sorry, go
uh,
ahead.
associate teaching professor. Yeah,
Associate teaching professor. And so, if this is your first program of professional previews, a little bit about this is that this is a chance for you all to sit down and ask questions and learn about the life of this professional and how they got to where they are in this STEAM career. And so, just a couple of ground rules. Remember to keep yourselves muted and your cameras off until the very end of the program. After we stop the recording, you will have the chance to unmute yourselves and turn on your videos and talk directly to Dr. Goller. Also, live captions are available in Zoom, so if you wanted to activate those, you can click on that at the bottom of the screen. And now we will pass it over to Dr. Goller.
thank you, and thank you for inviting me. I've really enjoyed working with you when you were here at NC State, and love the museum. And I am happy um we were able to reconnect. So where to begin?
Yeah, just start by telling us, a little bit about yourself and where or how you got to where you are in life. So, where are you from? How did you choose your current career path, and what is that career path?
Ooh, so I grew up in Mexico. So geographically, smack in the middle of Mexico. Mexico is almost like a boot shape. I am from Guanajuato, is the state, and it's northwest of Mexico City. So we are in the middle. We do not have beaches, but we have a really historically rich colonial city with cobblestone streets and cathedrals. And it was part of when Mexico was part of the Spanish Empire. It was a really important city. Um, and it it really changed how I interacted with people and learned from people, because luckily my dad, who was an American US citizen, um, US Navy retiree, retired in Mexico, loved coming to San Miguel even before retiring on it during winters, because we have fantastic weather and really awesome thermal hot springs. Um, And it's back in the 70s, it was a really popular place for uh, US vets, French Canadians, and Germans to retire down there. And interestingly, it has become one of the Travelers Magazine's fourth year in the row uh, best place to retire. So for me, it was great because I learned a lot from a community of retirees from different countries and different places. And my mom is Mexican and a Spanish and English as a second language teacher for grade school students. And she was actually this grade school coordinator when I was in that bilingual school, and did 40 plus years of teaching both in Mexico City and in San Miguel. And the reason why I mention all this is it was and it still is, a very artsy, um, a very colorful place to grow up in. And you were exposed to a whole bunch of different cultures, not only Mexicans, but also retired people that brought part of their culture. And some of the family friends were Germans, that and Canadians. And one couple, uh, he had been a prisoner of war in Germany for a long time and loved science and gave me a microscope. And along with my dad, whose brother was an engineer for NASA, and my dad was always very creative. I really loved uh, learning about stuff. So when I approached high school, I had two options: stay in town with lots of nightclubs and fantastic places, or go to the next state over to a science, technology, and math uh, high school with a university attached. In Mexico, the systems are very often you have a university system attached to a high school system if it's a private. And this one was a private one. At the time, they had 20 plus campuses. They had a bicultural program that they were testing out. That was half the classes in English, half the classes in Spanish. It was private. And I really wanted to do science and math. And my mom said, you can do that, but we can't afford it. Get a scholarship. And I was able to get a scholarship. And maybe my mom's mistake, but there was no dorm. So I lived with two college students, and that was fun. And one of them was a agronomist, biochemist, and gave me access to labs. And the other one was an accountant, but really into uh, calculations and models. And upstairs we had engineers. So I was lucky to, even as a high school student, be able to experiment in university labs.
Wow. Yeah, I bet that was really a really formative experience being able to have such early access to things like that. Have you ever come across anything similar to that in the States?
I I have I think the best school of science and math here in
Mm,
Durham is an example, because you have classes that are course-based research experiences, and at the high school level you're experimenting, you're testing things. And I was really lucky to be able to do that in Mexico in a really nice high school, um, uh, with a scholarship. Part of my wife and I were talking about this. Uh, Part of my scholarship, my work study, was based on cleaning the trackballs that we no longer have of mice in the computer lab for
yeah.
150 computers. The internet had just started and it was Netscape. And that was part of my job. And that gave me a 95% scholarship. And I
Wow.
was able to really learn about technologies, and also so both computer technologies, and learn a little bit about the lab by just hanging out with David, who was my Spanish roommate, who had access to a biochemistry lab.
Wow. And so, in your current field, what you currently do and study, I bet there was a lot of connections there from that early exposure to to those environments. And so, now you study all sorts of microbes and pathogens, right? So, what would you like to tell us about that?
Oh, there's a really cool connection. And at the time it was a little nerve-wracking. I've never been good in quotes. I've never been a good test taker. And in order to get into the high school, I had to do a standardized test. And I always mention this because I did the test, and I didn't hear back for a while. And then I heard back from someone saying, by the way, we had to grade your test manually because you skipped one of the bubbles. Early
Oh
on in the
wow.
test, I skipped one of the columns, and I was lucky to have some person and to this day I don't know, manually check my test and go, oh, okay, and then award me a scholarship. And the scholarship was for studying in this bicultural high school. It was an experimental program where they did math in Spanish one semester and then math in English the next semester. Probably not a, now that I think about it, I'm like, maybe that wasn't the best approach. But along there, I took a class called um, basically scientific methods. And as a high school student, that was a really innovative class. And at the time, there was an infection of Zika virus and Ebola virus going on. And I was really intrigued by Ebola viruses. And it was one of the early, all Ebola virus outbreaks are bad, but one of the early, really bad Ebola outbreaks, and people were using this thing called biotechnology to study the viruses. And I told my professor, I want to be a biotechnologist. And she's like, you do it. That sounds awesome. I believe in you. And she was a fantastic high school biology professor. Ironically, my guidance counselor said, biotechnology is not a major. We won't be able to offer you a scholarship because your aptitude test says that you will be a physicist. So you can either take the scholarship to be a physicist or find another place. And my mom started to panic because my dad had passed away, and my mom's like, you're turning down a scholarship because you want to study something else that may not exist in Mexico at the time. And I said, then I'll apply to US colleges that have biotechnology as a major. So I only applied to places that had biotechnology as a major. This is in the 90s. And instead of going to a big name school, I ended up going to a smaller institution called Worcester Polytechnic Institute that I had never heard about. I had never been exposed to uh, snow, first of all, in Western Massachusetts, but I picked WPI because it was project-based learning. And again, I had the opportunity to do research. And years later, I was really lucky to make that connection. WPI is now known for project-based learning with Christine Wowie's work, who was one of my professors. The person who said that that I should go after biotechnology ended up being in really high up in administration and student success at the level of multiple campuses for that campus system. My guidance counselor, who said biotechnology does not exist, ended up decades later opening a biotechnology program in that
Wow.
for the high school, a biotechnology concentration. And I ended up with this, at that time was really nearsighted or closed-minded. I was gonna go after the monkey that had Ebola or chase. I was gonna be CDC, Centers for Disease Control, a virus hunter. And that was my mission. And it was interesting because even when I went from undergraduate school to graduate school, I applied to a whole bunch of places, but my top choice was Emory because Emory was next door to the CDC. And I could be there. And I thought I would be a virus hunter. And I love viruses and bacteria and microbiology. But interestingly, and again, this is one of the fun things of life. You do rotations in different labs as a graduate student. I really wanted to join the lab of someone who is really well known for virus and vaccine like virus like particle and vaccine production for Rift Valley fever, Ebola, different viruses. So I did one rotation with that lab, one rotation with an influenza lab. And then my last one, I said, I really want to do something different, just to get experience, a bacterial lab, how bacteria attach to surfaces. And I really loved it because it included the computer side. It included
Hmm.
some of the genetics, and it was a really solid question. How do bacteria, in this case E. coli, attach to surfaces? So being open-minded is important.
Yeah, that's a very interesting niche, and science is kind of about finding that niche of what is this thing. We know this and we know this, but what is the thing that connects those two? So that's a question that I've never even thought of. How do these tiny organisms attach to different surfaces? Um, so yeah, that's an interesting question. Did you ever find out? Did the research ever yield any results? Did you know?
I got, again, really lucky. I joined this lab, even though I thought I was going to be a virologist. I joined Tony Romeo's lab. Tony had just moved from a UT, from one of the UTs, University of Texas. I love unboxing equipment. So I got to unbox and set up his lab with one of my best friends, Jeff Mercanti, who also joined the lab and is still at CDC. Um, and the graduate student that had traveled with or joined Tony from Texas and went to Emory had just done what's called a screen of E. coli to figure out which genes were involved in attachment. And Xin Wang had discovered a series of genes required to make a polysaccharide, a glue, basically the
Mhm.
sticky substance that makes E. coli attach. I had, and people were like, okay, done. Let's study PGA, this N-acetylglucosamine, this substance that helps E. coli and other organisms because it's conserved attached to surfaces. And again, Carlos had a really wacky idea. I said, well, we know the glue, but there must be an on and off switch.
Right.
And I bet that the on and off switch is in that collection of mutants that can't form a biofilm, that can't attach. I bet one of those mutants is not a mutant of the glue, it's a mutant of the on off switch. And I took a risk because people told me, well, all of the ones that can't form a biofilm or attach are probably uh, mutant E. coli that can't make the glue. And I was I convinced myself that there was some sort of connection between the glue to environmental signals, and it was somewhere in those couple of thousand E. coli mutants that we had in that collection. And I was able to find the E. coli regulator gene that turns on and off production of this glue.
So that's interesting. So the mutation only coded the switch and not necessarily for the production of this glue. Uh, how do you test for the stickiness? Like how can you tell if an E. coli is sticky or not? How does one do that?
Oh, this is a good one. So what Shin had done, and this was common in the field and in the early 2000s, it was really popular assay. George O'Toole was the one who created it, and I believe Roberto Coulter from Harvard. And O'Toole was at Dartmouth, and uh, Coulter is at Harvard. And it's an assay where you grow in a dish E. coli statically, or E. coli or another organism statically in liquid. You let it grow for 24 hours, 48 hours. Then you take the liquid out, you wash it a couple of times, and you stain with a purple dye whatever's left in the well. And whatever's left is what got stuck. So we were looking at purple means it was able to form a biofilm and attach. No purple means it wasn't able to attach.
Okay. Yeah, in my mind, it was a much less a much less intricate matter of sitting there under a microscope with tiny needles, just shoving shoving their little bodies and seeing, are they sticky or are
Are
they
they
not? Do they
sticking?
just slide
So,
around?
so interestingly, my first project, which didn't pan out but was a fun adventure, was when I first joined the lab. Tony got a fancy microscope that had an automated stage, and you could program it. And I've always liked the challenge of computer programming a little bit, and they're not really detailed coding, but simple scripts. And I was writing scripts to track bacteria as they swam to see when they would attach. To ask one of your questions, do they attach early? Do they not attach? How? And we had a fantastic researcher called Konstantin Agladze, who's now in Japan and has his own lab, but his background is physics. And he figured out that the bacteria form certain patterns before they attach on a slide surface. So almost like a dance to find where you're going to attach.
Interesting. So yeah, that's really cool. And again, questions that I never would have thought to have
Mm-hmm.
asked. Um, and so, how did your experience going to class and having this unique experience in high school and going to a completely new environment for undergrad and college, how does that experience how you teach your students today?
I think I am very, I've become very flexible, and I want to learn by teaching. And I'll say that that has changed in recent years as I get, get older and teach more. I think I think I'm not I shouldn't be the one just doling out information. I think we can co-create information. And that's what I like about open education, open pedagogy, citizen science, where we all contribute. And in some cases, in most cases, I think the questions should be driven by everyone, not just the person who has access to the lab. So I was always lucky that I had mentors, Tony Romeo for grad school, Pat Seed for a postdoc that I did. And then when I came to NC State, Bob Kelly here let me design a course based on microbial communities and identifying which microbes are there. And before that, at Duke with Pat Seed, Pat and I remember getting labeling tape or masking tape and setting stuff up to do this experiment and being really creative and having fun with it. So I think we can
Uh,
do that in the class, and it's more fun for me.
Yeah, and that means a little bit less time spending grading tests, right?
Exactly.
And actually transferring knowledge and not testing for memorization. Um,
Yes.
and so studying like infectious diseases, E. coli, that's one of those things that we all hear, and it's like, oh, don't handle raw chicken or don't eat, raw cookie dough because there could be E. coli in there. So you said that you were very intrigued by these infectious pathogens. What are some of the risks, and how do institutions navigate the risk of studying things like Ebola and whatnot?
Yeah, so I didn't end up studying like the really infectious viruses, though I almost, and friends of mine did it. There was a class, and I think there still is, that Emory offers, that's like the full suits and respirators, and learning how to work in environments, containment environments with proper air circulation and things. I never did that. I did work with bubonic plague, um,
Oh.
because it has the same genes required for making the glue to stick. And that was just me and being really careful and being working in the lab. And now I think more about safety in terms of, I'm really interested being in at NC State and having a vet school, composting. I'm very interested in sustainability. There are really interesting organisms in soil and in compost and in um, a, a farm uh, soils and farm waste, but they also have lots of not so good genes, or genes that may be good for them, but not so good for us. So we have really great technologies now to learn about the microbes without propagating them, by sequencing their genomes and not having to grow them in large amounts. And I think that's really exciting because that we can bring that excitement of discovering and learning about the mysteries of microbes to a teaching facility in a safe way.
Yeah, and I do love that added layer of safety that, new technology has allowed for. And I know one of the reasons why we like to have things like E. coli and other bacteria is because they reproduce very quickly, and their mutations, we can kind of watch any mutations and essentially an evolution happen in real time. Can that be simulated in a virtual environment? If we sequence the genomes, could we, you know, turn on certain genes or see what happens with random mutations in a computer simulation?
We can. And even just yesterday, I had a friend, I had the opportunity to bring a good friend, Ben Allen, uh, uh, and I had never met Ben in person. Ben gave a workshop and down the hill, and he is part of KBASE, KBASE.us. And that's a Department of Energy funded knowledge base. It's a web-based platform for bioinformatics and for analyzing, putting together bacterial genomes. It's mostly bacterial and plant and so some virus stuff. But what's neat, and we talked about in this workshop, is there are now tools to do what's called metabolic modeling. If you had the genome, and now we have literally handheld, the size of a cell phone, sequencers to sequence bacterial DNA. And
Wow.
students are doing it as we speak. It's working over there. Um, Once we have the bacterial genome, we now have the power of the databases, and we know the names of some of those genes. So in my case, I'm saying the chat, in my case, We knew NHAR was R for regulator, N for sodium, H for hydrogen. It was thought to be a exchanger of sodium. And that turned out to be the regulator for the glue. But we now know by having the database, the names and potential functions of most of the genes. Mind you, we even in E. coli, one of the best studied bacterial organisms, there's still a good chunk of genes that are Y genes that we don't know their name. They start with a Y. We don't know what they do. But with the genes that we have, we can take and the database information, we can take a new genome that students in this BIT 295 class are doing. And we can sequence that isolate that Amy Grundon's lab has shared with us. Bacteria growing in heavily contaminated soils. Why are they growing there? How are they growing there? We can sequence their genome, use K-base to predict what they are going to grow in based on their genes.
Wow.
So we can have it model growth in different, can it grow in heavily contaminated soils with heavy metals, for example?
Yeah, that's really that's really awesome that we're able to do this now. Um, and so I have a question that is kind of switching gears and kind of to something that you said at the beginning. You said that you grew up in Mexico and that your dad was American, your mom was Mexican, and you grew up in a very multicultural city or town. Are you multilingual? And if so, what languages do you speak?
Mostly Spanglish, but I speak English and Spanish. I grew up speaking both. Um, I joke around about it because I am um, probably one of few friends that had to take speech lessons to roll my R's, which was kind of fun. Um, but I can roll my R's. And I learned chemistry and math in Spanish. So I still do math and chemistry in Spanish.
Interesting. And so has this language difference, or you grew up bilingual, correct? Speaking both English and Spanish, right? Um, has that ever posed any barriers to you? Like, has that been the basis of any um, either discrimination or whatnot that you've faced? And what advice would you have for someone who doesn't have English as a first language trying to thrive in an American university?
That's a really good question. And I think there are barriers and there is discrimination, unfortunately. Um, I think often we our our graduate students and researchers are from all over. And
Mhm.
unfortunately, we often don't, at least I don't, and I should do this more. And there's really fantastic research going on in Spanish speaking countries. And we just don't access it because our go to is American science journals or European science journals or journals written in English. And
Right.
I think there are there is some really fantastic work being done that's not being connected. And it could be really unique samples not connected to exciting technologies we have. And language is a barrier. And I still remember this time. It was a conference in 2004, one of my first conferences presenting. And I was giving my spiel about E. coli, that I was still a grad student working on E. coli and biofilm formation when bacteria form a film or attach to a surface. And someone from the equivalent of the National Institutes of Health, but in Mexico came up and said, can you do this in Spanish? Well, I could not explain what I was doing in Spanish. And that, or I struggled. I ended up doing it, and that really made me think, I am losing something that is really personal and part of my identity. And from then on, I tried to uh, force myself to find ways of doing science in Spanish too. And just yesterday, I was talking to a company that does videos for and 3D, awesome 3D animations. And I'm like, my new pet organism, Delftia, Cedovorans, this bacterium that has some really cool properties, and it is able to take liquid gold and precipitate it into gold nanoparticles and maybe used for recycling our discarded electronics and recovering some precious metals. How can we do a video and include that explains an explainer video that includes English and Spanish tracks as well as uh, uh, captions? Or we were talking about American Sign Language. How can we make it so that it's accessible to more?
Yeah, so I that was going to be the other question I ask is on the flip side of that, how has being multilingual been an advantage? So being able to go into those spaces and have those opportunities to talk about all the amazing things that you do, but in a completely other language. Um, like I remember, and I think about this all the time. Sofia Vergara from Modern Family,
Yep.
she there's this quote where she says, "You have no idea how smart I am in Spanish." You know, everyone, they make fun of her accent. They say, "Oh, she's just dumb," and blah blah blah. But it's really hard, thinking about that. If you've learned a second language very later on in life, you don't have all the nuances that you would if you grew up with it. And so um, that is something that I think about a lot and really have taken to heart. Of I've worked with many people from all over the world, and of course they might struggle if English is their second language. They're coming to an American university and trying to speak English. You already know another language more than I do. um, and so yeah, there are these scientific articles, I'm sure, that are written in Spanish that American professors or universities or scientists have just never even been able to access. Um, I'm sure there's Google Translate, and we can try to try to do that, but it can. Yeah, I'm sure there's some gaps that have already been bridged. It's just they haven't been bridged in English.
Right. And there are some really cool opportunities. Exchange programs, study abroad. I'm excited to be able to do something like that in the coming years, hopefully.
Yeah, and so you were mentioning your little favorite pet microbe, Delftia, and that it can precipitate gold. So can you go on about that? Like, where is it found, and how did you find this thing?
I was telling these videographers yesterday, and I was actually telling them in Spanish. Um, it's a really fun story because I did after Emory with Tony. I really wanted. I've always had a public health side. And I kept on nagging Tony that I really wanted to do this beyond the lab E. coli strains. And one day he said, so just take a public health class and learn about that. And I'm like, that's true. The School of Public Health, the Emory Rollins School of Public Health is there. So I ended up taking a School of Public Health classes. I, I think I did it for 4.5 years.
Wow.
At night classes, morning classes, and my boss, my mentor, Tony, was okay with it. And I'm mentioning that because it's uh, a way of saying don't stop learning and take advantage of the opportunities you have. Public health wasn't something that may have directly helped me in the lab with my research on E. coli, but it really broadened my perspective. So when I went to Duke, I worked with Pat Seed, who was an MD, is an MD PhD. We really wanted to look at how can we find compounds that drugs to break up the interaction between pathogenic E. coli that cause urinary tract infections and the cells or the surfaces. Because if they get stuck or if they form intracellular, so inside the cell, biofilm communities of microbes of E. coli stuck together, you get persistent UTIs that are recalcitrant, so harder to get rid of with antibiotics. So my public health background and Pat's medical um, knowledge really helped us connect clinic to wider needs and look for that, look for potential solutions. So when I came to NC State, I could not bring my UTI causing agents here because in a teaching facility that would not work. And I was hired to teach this metagenomics class. And by metagenomics, we mean meta, lots, and genomes. And we now have the ability to take a swab a surface, and without growing all the organisms that are on that swab head, break them open, get their DNA, and use those databases I've been talking about of sequences to learn which microbes were in there, in that surface or on that, in your gut, for example. And Rob Dunn has always been someone I look up to. And we were actually having a conversation when I was designing this class in 2012. And he had just finished the citizen science study engaging households in sending swab samples from the pillowcase, toilet seat, entry door, kitchen counter. And I maybe jokingly said, well, have you done the kitchen sink? And we both stopped and we're like, the kitchen sink is a great place for biofilms. You got the running water and the bacteria that are there have to be resistant to flow of water, have to be able to stick there, and have to be able to survive on like a metallic surface or goo. I wonder what's there. So as part of this class in 2013, we started swabbing kitchen sink drains and learning what microbes were there. And the first semester I did the class, someone's sink had 60% Delftia acidovorans. And that's Delftia acidovorans. And I had never heard of this organism. I'm like, why does this sink have that organism? And we Googled it in front of the class. And the first article was that same semester, they had published, scientists in Canada, researchers had discovered that Delftia is able to take liquid gold and it produces a tiny protein that it sends out, that secretes. And gold and other heavy metals are usually toxic to bacteria. And what Delftia does is instead of spending energy pumping it out, which most other organisms do, it produces a tiny peptide, a tiny protein that takes gold and coordinates it. Basically, liquid gold captures it, gums it up so that you get gold particles. So it basically poops gold.
Wow.
And I've been obsessed ever since. because it's a really neat story of bacterial magic.
Yeah.
And it's an organism that's all over. It was in Matt's sink because Matt was washing vegetables and it was probably in soil. And nowadays, there's a company in New Zealand that's using this organism and another one to recover gold. It's called Mint Innovations, to recover gold from discarded electronic waste, because some of our USB cables and circuit boards have gold coatings.
Yeah. And that, the hearing about how prolific e-waste is, and especially when the cell phones and computers, they come out with a new version every year. And
Yep.
what happens 99% of the time? People in either end up, just leaving their phone in their drawers and it just sits there for years, or it gets tossed out, or they get traded in. But then what happens after that? Are we able to use those components and whatnot? So that sounds like what you were going into, of how we can use some of these microbes to recover those precious metals and reduce our waste.
And I love the history of Delftia because it's called Delftia as a tip of the hat, acknowledging the historical microbiologist from Delft, Netherlands.
Ah,
So Leeuwenhoek was the one who played with the microscopes, fine tuning it.
mhm.
And likely one of the first things he saw was like uh, seeing uh, stuff on people's teeth, and he did sweat. But he also did soil and water, and he may have seen Delftia without knowing.
Wow. Yeah, I mean, it makes sense that these bacteria have been around for hundreds, thousands of years, right?
And we just didn't have a name for it till two decades ago. So it was not a named organism. It had different names, and then people realized with the uh, progress we've made with sequencing and learning about microbes, it started to, it was Pseudomonas acidophorans, Comamonas, and then people figured out, oh, it's a branch on its own. And so for the last couple of years, I've been maybe obsessed, but just curious, and bring students in the process of, let's figure out, what do these organisms do. So we've been getting bacteria from the USDA and labs in Canada, Delftia, and then sequencing them and seeing, do they all have these gold scavenging genes? And so
Wow.
far they do.
Yeah. And so if your abuela were here right now with us and didn't hear the rest of this, if your grandma were here, how would you explain to her what you do now?
Oh, that's a good one. I would say hopefully I'm an educator, and I come from a family of educators. My mom was a teacher. My dad was but I didn't say, my dad taught people how to land on aircraft carrier. And
Yeah.
from my mom's side, they were jewelers and teachers. And I hope that I'm I'm my partner and her mom is a teacher. I hope that I'm an educator who shares some passion for figuring out how we can learn more about the microbes around us using cool techniques.
Yeah. Yeah. Well, that's a very nice, sweet way to put it. I know a lot of us scientists and researchers, we're great at explaining what we do to other scientists and researchers, and I've had this own experience with my own grandma of
Mhm.
she's like, "Oh, what are you going to school for?" And I just say, "Biology." "So you want to be a doctor?" "No." Not quite. And so that's why that's a question that I try to ask most of our professionals is how would you explain what you do to your grandmother? Someone who probably doesn't quite understand what you do, but full-heartedly supporting you
Right.
and doing great.
Yeah.
So thank you for that explanation. Um, and so now are there any like closing words or final words that you wanted to impart for everyone watching?
I think we often think of linear career paths, and mine was circular. And I
Mm,
think everyone else uh, has different career paths. So circular in the sense that I really wanted to study biotechnology and major in biotechnology. And now I'm in a biotechnology program teaching biotechnology to others. And that's something I wouldn't have thought about. And maybe my guidance counselor was right in a sense. There wasn't biotechnology as a major back then, but uh, it was a set of tools. And I think along your career, you find a set of tools to learn about questions that interest you. And I would
yeah,
encourage people to maybe find connections or find ways of using some of those tools.
yeah. Well, that's great. And so thank you so much, Carlos, for sharing all of these amazing things that you've done. Um, are you well, I guess are there any questions from anyone in the chat? I know we had a question here. Have you ever used a field microscope before? And what is the coolest microscope that you've used?
Oh, so I got as a present a microscope when I was probably 6 or 7, and I remember playing with it and playing with the mirror. And nowadays, microscopes are so cool. Uh, I took as an undergrad a class, and this has nothing to do with viewing microbes, but as an undergrad, I took a class where we used atomic um, or electron microscopes. And it was a really cool class with just a handful of students. And I remember going to the lab and there was pollen, and I captured some pollen and was able to view it as really awesome structures on this extremely powerful microscope. And I loved that. There was a scanning electron microscope as an undergrad. That was really cool. And nowadays, I don't get to play with microscopes that often, even though we have some cool ones. And in an hour or two, a fantastic undergraduate uh, is going to challenge me, and we're going to look at some Delftia under a microscope with some fluorescent stains.
Ooh. So they'll be nice and wonderful colors,
Green,
yeah? Yeah.
yes.
Yeah, and so another question that we had, you mentioned virus hunting or that term, being a virus hunter. You said that you didn't opt to go down that career path, but do you know a way that someone could pursue that? It's-
Think I'm actually teaching a class that I love right now, first time, but it's an honors class on outbreaks. And we were just talking this week about how the Centers for Disease Control and other public health agencies have um, disease surveillance or public health jobs that use molecular techniques or use mathematical modeling to study outbreaks. And in that case, you are still a virus hunter.
Okay. So yeah. Well,
And in a
I think
way,
we're. Oh, go ahead. Sorry.
oops, in a way, I think I'm still a virus hunter because when I swab something and we're looking at what microbes are in there, we often find a virus DNA
Yeah.
from viruses.
because they're everywhere and we don't even recognize
Yes.
it,
Yep.
and they're super small.
Hmm.
Uh, yeah. Okay, well, thank you so much, Carlos, and everyone else for joining us today. I hope that you all learned something and got some valuable lessons. Um, we'll be ending the recording soon, but if you wanted to hang on for just a second and say hi to Dr. Carlos or ask him a question directly, we'll have a few more minutes for that. We're a little bit over time, but that's okay. And so otherwise, you can join us next month for hydrology with Dr. Brittany Hunt. And also, the museum is having a SciTech Expo on April 22nd. So Earth Day, come on out. It will be an all-day event. We'll have all sorts of tables and scientists and researchers and activities for the entire family. So thanks everyone for joining us and hope to see you all next time.